In the last video, we discussed the major structural features of DNA. The topic of this video is the differences in structure between DNA and RNA and some of the functional implications of those differences. So to start off, let's just compare and contrast RNA and DNA. So first, the compare. Both of these are polymers of nucleotides. So they're both built out of the same building blocks. But even though they're both built out of the same building blocks, there's still a lot of differences between them. The first one is that RNA uses uracil instead of thymine. RNA is also single-stranded, which is really important because it allows the RNA to be able to adopt a variety of structures that allow it to carry out a wide variety of functions. So RNA is involved in carrying and decoding the genetic information during protein synthesis. It's involved in catalysis, and it also plays roles in regulating gene expression. And this is possible because RNA is able to base pair with DNA, and it's also able to base pair with other RNA molecules. And that allows it to play this regulatory role during gene expression. RNA also has a 2' hydroxyl group because it has ribose sugar instead of deoxyribose that contributes to RNA being much more unstable than DNA. So when you think about it, DNA is the genetic storage molecule. It has to be around for a long time to be able to be passed on to the next generation, and it has to be intact when it's passed on. So DNA has to be very stable. RNA, on the other hand, is not as stable as DNA due to this 2' hydroxyl group. So let's talk about this in a little bit more detail. So first difference, we said that that RNA uses uracil, and uracil is just going to replace thymine. It's going to base pair with adenine. And the reason why this happens in RNA and not DNA is related to some of the proofreading mechanisms that DNA has. Since RNA is a temporary molecule, it's just produced to carry a message and then degraded. It's not as important for it to be exactly perfect. It doesn't need all these proofreading mechanisms. In DNA, thymine helps to ensure that the DNA is being properly copied every time. It provides a place for proofreading mechanisms to be used. Okay. Now, let's talk about how RNA, being single-stranded, can be really important for its function. So, because RNA is single-stranded, it can actually fold in on itself and have these base pairing interactions between nucleotides within the RNA molecule. So this gives rise to a lot of different structures that RNA can adopt based on how it's almost folding on itself. So this is called a stem loop. It's one of the simpler structures. It's very common. A lot of the more complicated RNA structures have stem loops in them. They can also form very complex structures. So like tRNA here, or this is a ribosomal RNA from a human. And you can see that some of these RNAs can be quite large and have quite complex structures. And this allows RNA to carry out many different functions. So one thing that you'll start to notice in this class is that there's this guiding principle in biochemistry that form or structure follows function. So what that means is that in order for a molecule to be able to carry out a certain function, it needs to have a certain structure. So because different RNA molecules can adopt quite different structures, that allows them to be very versatile and play all of those functional roles that we talked about in a few slides ago. It's a very versatile molecule, more possible functions, needs to have more possible structures. So that is how RNA being single-stranded can be really important for its function. But we also said that despite this versatility, and possibly a necessity of that versatility, RNA is very unstable. And the major reason for this is the presence of this 2' hydroxyl group, because that 2' hydroxyl group leaves RNA very susceptible to hydrolysis, specifically base catalyzed. So what can happen is that a base can come and deprotonate that hydroxyl group, creating an oxyanion, which can act as a a negatively charged oxygen, which can act as a nucleophile and attack this phosphorus atom in our phosphate, resulting in, at one point, this intermediate where our phosphate is bound to all 5 of these oxygen atoms. Then the oxygen connected to this sugar here detaches, resulting in basically cutting that molecule into 2 pieces. So the one piece ends up having this cyclic phosphate group here, and the other group has whatever is left of the chain on the other side. That RNA strand has now been cut in half. It's no longer carrying the same message. So this 2' hydroxyl group makes RNA very unstable. Whereas DNA, not having that 2' hydroxyl group, doesn't have this problem where it can have self-hydrolysis. So that allows DNA to be very stable, last a long time. Whereas RNAs, they're temporary. They're recycled very, very quickly. So being easy to degrade helps with that part of their role in the cell. Also, being easy to degrade can help with fine-tuning gene expression. Because the cell can very quickly start degrading all of the RNAs that make a certain protein, resulting in less expression of that protein, allowing it to regulate the activity of the cell. Whereas DNA, its entire job is to just sit in the nucleus and last a long time and not get degraded so that it can be passed on to the next generation intact. So DNA needs to be very stable. Okay, so that's a little bit about how the different structures of DNA and RNA can be very important to their functions in the cell. In the next video, we're going to talk about how base pairing is an essential part of the central dogma of biology.